Mm-hmm.
Yes, please.
Okay, so my topic was sending samples to the lab, and it had, as part of the question, pediatric tumours, sarcoma, oriented specimens like Mohs, and eyelid and conjunctiva. So I looked through all the kind of past papers about sending specimens to the lab. Interestingly, I didn't really find any questions about sending pediatric tumours or sarcomas, or any past questions about Mohs procedures in the past question bank. And I found that questions about sending specimens were only usually like a subsection of the question. The questions also would include, you know, either describing the clinical features of the relevant condition that you were sending the specimen for, or coming to the diagnosis based on a clinical vignette and then asking how you would send the sample. And then there'd always be another part about when the specimen actually got to the lab, what would you do, or like how would it be processed, and what you'd actually order, or what results you would expect. And then some of them had, you know, wanting you to describe also the histopathological features of that condition. So it didn't seem to be that common. I mean, there were some questions about sending specimens to the lab, so I do think it is important to know. The most common question that came up in the past papers was about lacrimal gland biopsy for suspected lymphoma and how you would send this specimen. So that came up a few times. There were also questions to do with kind of more microbiology, so sending ocular tissue or fluid for PCR, what kind of samples you would send and how you'd send them. Also similarly for endophthalmitis and acanthamoeba. There was quite an old question from 2004 about a skeletal biopsy. The other questions that came up were just a few times were how to orientate and send a specimen when you're looking at OSSN and sending a conjunctival specimen. There was one question about how to do cytology and how to send that specimen. And then the other areas were ocular cicatricial pemphigoid, sending a temporal artery biopsy, and then just how to send tissue for cytometry. So those were the main kind of areas that past questions looked at for sending lab specimens. Overall, the questions were quite straightforward to understand. Just there was like two that you know, just a little bit of digesting. So this one said, describe the range of specimens and laboratory tests which may be employed to investigate a lacrimal gland biopsy to suspect lymphoma. So when I first read this, I was thinking range of specimens, what do they mean? I found this a little bit confusing, but I think that they were, you know, meant, you know, a needle biopsy versus a complete excision. But then I thought, could this also be code for, you know, how are you sending the tissue, like fresh versus formalin? So I think you probably need to discuss all of those issues. And then the laboratory tests, I think that they wanted you probably to talk about would be flow cytometry, H&E, immunohistochemistry, and cytogenetics in this kind of question. And I think for a lot of these sending specimen questions, I think it was important to talk about discussing it with your pathologist in advance so that the lab was ready and expecting a sample. The other question that also was maybe not answered well, according to the examiner's feedback, was discussing the steps involved in assessing surgical margins intraoperatively when excising a BCC of the medial eyelid by both the surgeon and pathologist involved. So I think they really wanted you to talk about frozen section, and that's what the pathologist would consider to be intraoperatively management. So again, just talking about the communication and how you would send the specimen. Yeah. So I chose a past question from 2010, but this question also did come up again, a similar question in 2016. So it was more a clinical vignette, 72-year-old European woman presenting with several months of gritty, painful eyes, painful mouth, difficulty eating. On examination, you noticed subepithelial scarring with shelling in the fornices, formation of fibrous bands with symblepharon, and then her mouth's also involved with a blister and gum line. So I had a go at answering this question, and overall I feel like I managed to answer the main points of the question. The diagnosis was obviously ocular cicatricial pemphigoid. I found that the model answer was very good. It had a lot more detail than I had, so I think it's just a matter of practicing and learning all those details for the exam. I did, however, in the model, well, not the model answer, but in the notes available online with the way the questions are, I wasn't sure if there was a mistake. So basically, the key point for the question was sending fresh tissue for immunofluorescence to make this diagnosis. And the notes talked about sending fresh frozen tissue for immunohistochemistry and immunoperoxidase. Well, I understood that immunohistochemistry can be performed on tissue that's been fixed. So I think that the fresh frozen tissues for immunofluorescence, which is different to immunohistochemistry. That's my understanding. Correct me if I'm wrong.
You're right. You're right. You're getting it perfectly. Yeah.
So yeah, for this question, I think the important points were just to be aware that for this condition, you can actually send oral biopsies. Even though we're kind of always focused on the eye, there's a lot more tissue in the mouth. So you can still come up with the diagnosis from the oral biopsy. And things that I wasn't aware of was that you do need to take biopsies from 2 sites, and that even if you send a biopsy, you might still not get the diagnosis. So a negative biopsy doesn't exclude the diagnosis. And the sensitivity can be as low as 50%, especially if disease has been long-standing, because you get destruction of the basement membrane and you can lose that characteristic pattern of immunofluorescence where the basement membrane lights up, showing the complement and the antibodies. Again, talking to the pathologist in advance is important so they can be ready for your tissue and process it in a timely manner. So top tips for these kind of questions, I just think it's important to not forget to mention communication. I think that's important to actually get a proper diagnosis and to get the tissue dealt with in an adequate manner. Also including adequate history and information on your request form so that the pathologist knows what you're thinking, what diagnosis is likely, and they can run the appropriate tests. And then on another note, for the lacrimal mass questions, I just learnt that if you see a whole capsule in imaging, you should send the specimen whole. Perhaps it's contraindicated because you might be seeing a pleomorphic adenoma and increasing risk of recurrence, as opposed to the lacrimal biopsies where you would be doing lacrimal biopsies when there's no capsule on imaging and you see destructive disease and you've got an unclear diagnosis. So looking at things like lymphoma, again, you're sending fresh tissue as well for things like flow cytometry. Yeah, so that's my presentation.
Okay, thank you. Should we pop back a couple of slides where you said that you didn't understand the questions that was being asked? So the bottom question there, you said there was no Mohs questions, but I think that the bottom question there is a Mohs question.
Oh, okay, there you go. I didn't understand the question at all then.
Because that's when we assess the margins interoperatively, yeah?
Oh, I thought it was just about sending the tissue, the frozen section. All right.
So if there's a BCC, you can send the main specimen and get a diagnosis of a BCC. But my understanding of Mohs is that you want to assess the margins interoperatively to do the minimal excision necessary. Yeah. So that's what that was about. So anyone who's hopefully been in a Mohs situation will understand what you have to do to send it properly, as in it needs to be orientated and marked and tacked down so that it doesn't curl up and get all distorted as you send it. So because we really want to embed it the right way. So if you're doing a Mohs, we want to know what's the inside and the outside surface, yeah, so that we can embed it, so we cut down to the outside surface so that we can see that that margin is clear. Would you say that was important, Amanda?
I think, yes, the key word that I see missing from your answer was orientation, which is what Diane has expanded about. We want to know which is the critical margin and which is the further away margin. So orientation, there's many ways to do that.
And to be honest, pathologists like putting these questions in because it just drives us nuts when we can't give the right diagnosis, any diagnosis at all, because the wrong specimen or information has been sent. So in the top one, where we're describing the range of specimens, so let's think of what range of specimens you would send to the lab. You could send an FNA, you could send an incisional biopsy, you could send an excisional biopsy, and you would probably want to send it fresh. In the sarcoma questions, so all pediatric tumours need to come fresh because their biology is driven by mutations, and it's the same with sarcomas. So always send peds tumours and sarcomas fresh. But importantly, please give a history. Give a history of previous treatment, give a history of what you suspect the clinical differential diagnosis might be, because it's the intersection of the clinical diagnosis and the histopathologic findings that often give the clue to what the true diagnosis is.
To expand on the communication, which is a very important part of the answer, the important part of the communication is not to send the fresh specimens after hours and the weekend.
Yeah, we are not here.
No, and our technical staff are not here, and the flow cytometry people have gone home. So 4 o'clock on a Friday is just going to sit in the back of somebody's fridge until Monday. So timing is critical. That's a subset of the communication.
So basically that means that if you are scheduling a query lymphoma, please schedule it in the morning. Yeah.
And that's what the communication is, because we'll tell you again.
Yeah, because we need to, the latest we can usually get it in the lab is 4 o'clock, and then you've got to have lead time where people carry it over there, and then you've got to start the operation. So if you're starting at any time in the afternoon, there's a risk it'll get there too late.
Because the crux of this thing here is we are trying to get additional information, not just diagnosis where you could just slap it into formalin, but additional prognostic information, which requires these special tests and things, which require the fresh tissue. Because usually you kind of know the diagnosis is going to be a lymphoma, but you want to know what sort of lymphoma and what grade it is. And those require more than formalin fixed tissue. That's why you need the fresh.
Okay, should we do the closing survey now? I'm
yes,
not quite sure what you put in there, Amanda, but I do know I've put a draft schedule up for the 24th next week. And there's a little space at the end that I'd really like to fill in with things that you desperately want to know. So next week's mostly going to be on lids and cornea. But if there are burning issues, please identify in this survey. Next week, I would like to cover, and we'll see if we can manage it.
Can we just spend a second on fresh samples and
go for it?
Why, from your point of view, it's so important?
Okay, so there's a number of procedures that we need to do in the lab that we can only do on fresh specimens. Flow cytometry. So what happens in flow cytometry is they send the tissue down to the flow cytometry people who homogenize it. They separate all the little cells out, which are then stained with fluorescent markers and dripped one by one through a narrow tube. And very clever sorting devices then count them and split them. So we can't do that on fixed tissue. The second thing is all about genetics. Okay, so while we can do FISH on fresh tissue, that will identify known translocations and things that we're looking for. It's actually really helpful if we can have a carrier type just to grow the cells, do a metaphase spread, and find anything else that's out there. And we can look at the genes quite at close range now, doing what's called a microarray, and we can get a really good look at what's happening in relatively small segments of chromosomes. So flow cytometry and genetics are the most important part for tumours. For pemphigoid and other blistering skin diseases, we need it for immunofluorescence because that only works on fresh tissue. Anything else to add, Amanda?
It's, yeah, fresh is very useful, but we have to be there to receive it and triage it. And then, you know, it's not your problem anymore, it's our problem. It's just your problem to get it to us in good condition and good timing. I think also do the survey, but there's no extra box. The place to put your requests for next week, I think, should be on the bottom of the 17th of October Google Doc. How about that, Diane?
Or how about just in the messages?
Yeah, or in the messages.
Yep. Yeah, that would be, yeah.
Okay, so do your survey and then come back and put your things in the messages. Survey's down there. It's anonymous. I don't know. We don't know who you are. Turn your video cam off, whatever, and then we'll turn it back on when you're finished.
Back, yeah.
It'll take between 3 to 5 minutes.
Amanda, I couldn't find your frozen section video, which I know that you've made. After your frozen section, I think it was on Vimeo one time I saw it.
Oh, frozen section of how to do a frozen, the technical part.
Yeah, what happens during a frozen section?
Oh yeah, that was from Yale Medicine. It wasn't mine, but I'll put the link on the Google Doc.
Yeah, that would be great because I've got the Wills Eye Hospital one, and
yes.
I'm preparing iris conjunctiva thin specimens, and I've got tissue processing, but I don't have frozen section there.
Yep, no problem. I'll just see if I can find it now.
Thanks.